Hello everyone and welcome to this series of amino acids, and this is the first video, and in this video we'll learn about amino acids, general structure of amino acids and their types, which includes alpha amino acids, beta amino acids and gamma amino acids, and finally we will learn about the stereoisomerism in alpha amino acids. So let's start with the lecture and see what is the general structure of amino acid. So every amino acid is made up of 5 different components, and the center is having a carbon which is attached to 4 different groups. So we have a hydrogen, we have an amine group, we have a carboxyl group and a R group attached to that. So when a carbon is attached to 4 different groups, they are called as chiral carbon, or the center is called chiral center. So this carbon here is chiral carbon, and that's why they are called as alpha carbon. So this alpha carbon is the carbon which is attached to 4 different functional groups, and that's why they are called as amino acids, and the type is alpha amino acids. Let's understand what is an amino acid means and what is their role in the polypeptide. So each amino acids are attached with each other and finally forming the polypeptide. So if you have a polypeptide of supposedly 5 amino acids, each one represents amino acid 1, amino acid 2 and finally amino acid 5. And each amino acid are attached together with the help of a bond called peptide bond. So this is general structure of amino acids having 4 different functional groups attached to them, and this carbon is called alpha carbon. And this is a structure of polypeptide which constitute the amino acids. Let's move on and understand the types of amino acids. It could be alpha amino acid, beta amino acids or gamma amino acids based on attachment of NH3 plus, which is amine group. Okay, so we will understand how amine group is attached in general structure of amino acid, giving three different types of amino acids here. So we have to understand what is the priority functional group you have here. In this case, the priority functional group is this one, which is carboxyl group, and this functional group is your priority. So any carbon attached to this functional group will be called as alpha carbon. So this is called alpha carbon, and there is no other carbon. So in this amino acid we do not have any alpha carbon. No, only alpha carbon. This has only alpha carbon and no beta carbon. So this amine group attached to this alpha carbon, and that's why this is called as alpha amino acids. There is no beta carbon here. But if you see the second example, you have functional group which is carboxyl group here, and then you have a carbon attached to that called alpha carbon, and you also have a second carbon attached to that which is beta carbon, and your amine group is directly attached to your beta carbon, and that's why this type of amino acids are called beta amino acids, because your NH3 is attached to the beta carbon of the amino acid. Here comes with the third example having carboxyl group, and in this functional group it attached to the first carbon called alpha carbon. This is beta carbon and this is gamma carbon. So your amine group is attached to the beta carbon, and that's why they are called as to the gamma carbons, and that's why they are called as gamma amino acids. So I think at this point we have clear about alpha amino acids, beta amino acids and gamma amino acids. Let's understand what do you mean by stereoisomerism in alpha amino acids. So stereoisomers basically means if you put a mirror image between two molecules, supposedly L amino acid and D amino acids, you can definitely see a mirror of each functional group: carboxyl group, hydrogen, NH3 and R group. So when they have mirror of each other in their attached groups, then we call them as stereoisomers. when the mirror images are non-superimposable, which basically means your left hand or right hand cannot superimpose to each other. If you draw your left hand here or right hand here, it will not superimpose. So these mirror images are not superimposable mirror images, not non-superimposable mirror images. And as we see, they are type of isomers which has non-superimposable mirror images, and we call that a different term which is enantiomers. So most of the amino acids which has two non-superimposable mirror images are enantiomers, and they have an exception, and the exception is glycine. Why? Because glycine has H in place of R, which basically means when you draw general structure of amino acid with COOH, this is amine group and this is hydrogen, and you have a R. So in place of glycine, your R is H, and this H is hydrogen, which is very similar to this. So this carbon is no more chiral. This is not chiral, and this is the only amino acid which is optically inactive. All other amino acids are optically active, basically means they have a chiral carbon, except glycine, which doesn't have a chiral carbon. This is all for this video. Stay tuned for the next video. Thank you so much.